Characteristics and classification of living organisms. In this chapter you will learn about the 7 characteristics of living organisms. Find out how the binomial system is used to name organisms. Practice using and constructing keys. Describe how to classify vertebrates and arthropods. Describe the features of the 5 kingdoms of organisms. Describe how to classify ferns and flowering plants. Outline the features of viruses. Getting started. 1. The list below contains some features of living organisms. With your partner, discuss which of these features are found in all living organisms. Breathing, excretion, a blood system, a nervous system, sensitivity, growth, reproduction, movement, nutrition, respiration. 2. When you have made your decisions, write a very short description of each of the features you have chosen. Be ready to share your ideas. The puzzle of the platypus. In 1799, a dead specimen of a strange animal was taken to England from Australia. The animal had a beak and webbed feet, like a duck. It had fur, like a mole. No one knew whether it laid eggs or gave birth to live young. So, was it a bird? Was it a mammal? No one could decide. It was studied by Dr. George Shaw. To begin with, he thought it was a hoax. He looked to see if the beak was stitched onto the head, but no, the beak was clearly a genuine part of the animal. Dr. Shaw gave the animal a Latin name, Platypus anatinus. Platypus means flat-footed and anatinus means like a duck. However, someone then pointed out that the name platypus had already been taken and belonged to a species of beetle. So, another name was suggested, Ornithorhynchus paradoxus. The first word means nose like a bird and the second means puzzling. The name has now changed back again to Platypus anatinus. Figure 1.1. Platypus is adapted for hunting prey underwater. Later, proof was found that platypuses lay eggs, rather than giving birth to live young. However, they feed their young on milk, which is a characteristic feature of mammals. Scientists eventually decided to classify the platypus as a mammal. It was put into a new group of mammals, called monotremes, which also includes the echidnas. Discussion questions. 1. Scientists give every species on Earth a 2-word name that is used by everyone, all over the world. Do you think this is a good idea? Why do you think this? 2. Scientific names for organisms are in Latin, which is a language that no one speaks now. This naming system was invented in the 18th century. Do you think using Latin is a good idea? 1.1 Characteristics of organisms. Biology is the study of organisms. An organism is a complete living thing such as yourself, a platypus, a bacterium, or a mango tree. There are very many different kinds of organism on Earth, but all of them share 7 characteristics. Some non-living things have some of these characteristics, but no non-living thing has all of them. Movement is the ability of an organism, or part of it, to change position or place. It is easy to see most animals moving, but less easy to see a plant move. Figure 1.2. Characteristics of living organisms. A few plants can move parts of themselves quite quickly. And in almost any plant, if you look at the cells under the microscope, you can see chloroplasts moving about inside them. Figure 1.3. This fly is about to be caught in a trap. The Venus flytrap leaves will close together, trapping the fly inside. The plant will digest the fly and absorb nutrients from it. Respiration is the way that organisms obtain energy from nutrients, usually from glucose. Respiration is a chemical reaction that happens inside every living cell. You will find out much more about respiration in Chapter 11. Organisms use the energy that they obtain from respiration to make other chemical reactions in their cells happen. All of these chemical reactions, including respiration, are called metabolism. Sensitivity is the ability to detect changes in the environment and respond to them. These changes may be in the internal environment or the external environment. For example, you use your ears to detect someone talking to you. Plants detect the direction that light is coming from and can turn their flowers to face the sun. Growth can be defined as a permanent increase in size and dry mass. All organisms can grow. Some organisms, such as a pufferfish, can get bigger just for a short time to help to deter predators. This is not growth, because the fish goes back to its original size when the threat has gone away. Plants and animals grow by producing new cells. If you are studying the supplement, you will find out how they do this in Chapter 16. Figure 1.4. When a pufferfish detects a threat, it swallows water and inflates its spiny body so that predators cannot easily eat it. We can measure growth in many ways. One method is to find the dry mass. This involves finding the mass of several organisms of the same type over a period of time. An organism is killed and dried. The mass of its body without any water is then found. After a particular period of time, for example, one day, the dry mass of another of the organisms is found. This is repeated with several organisms for the growth period being observed. Reproduction means making more organisms of the same kind. Not every individual organism can reproduce, but at least some individuals of each kind of organism can do so. You can find out more about reproduction in chapters 14 and 15. There are many different chemical reactions going on inside every living cell. Some of the products that are formed in these reactions are not needed by the cells. These waste products are removed from the organism in a process called excretion. Excretion also removes substances in excess of requirements, for example, extra water. You can find out about excretion in humans in Chapter 13. Nutrition is the last of the 7 characteristics of all organisms. All living organisms need chemicals to build their cells, and also as a source of energy. Nutrition means taking in materials that provide these things. Plants and animals have very different methods of nutrition, and you will find out about this in chapters 6 and 7. Activity 1.1. Matching the characteristics of living things with their descriptions. Work in a group of 4 or 5 for this activity. You will need 14 pieces of blank card, all exactly the same. 1. Write the 7 characteristics of living things on 7 of the pieces of card. 2. Write descriptions of each of the 7 characteristics on the other 7 cards. 3. Shuffle each set of cards. Place them face down in 2 rows of 7. 4. One person then selects a card from each row and turns them face up. If the name and description match, this person keeps the 2 cards. If they do not match, they place them face down again in the same positions. 5. Now the next person does the same. 6. Keep taking turns until all the cards have been taken by someone. The winner is the person with most cards at the end. 1.2. The biological classification system. Classification means putting things into groups. There are many possible ways that we could classify organisms. For example, we could put all the animals with legs into one group, and all the other organisms into different groups. But this would mean that insects went into the same group as vertebrates. This would not make much sense, because we can easily see that insects and vertebrates are very different kinds of animal. Figure 1.5. Although they both have legs, these 2 animals are not closely related. The ant is an insect, so it belongs to the arthropod group. The chameleon is a reptile, and belongs to the vertebrate group. Biologists try to classify organisms according to how closely they think they are related. Long ago, perhaps 4.5 billion years ago, the first living organism appeared on Earth. It would have been a single cell. Since then, over a very long period of time, this cell gave rise to more complex organisms. For example, We think that all mammals descended from a species that lived more than 200 million years ago. This species was the common ancestor of mammals. All mammals are related because they all share a relatively recent common ancestor. When we classify organisms, we look for features that they share with others, which suggest that they are related to one another. This is a useful way of classifying things, because it helps us to understand how an organism works. If we find a new animal that has hair and feeds its young on milk, we know that it is a mammal. We already know a lot about it, even before we can study it in detail. Species. The smallest group into which biologists classify living organisms is the species. A species is a group of organisms that can reproduce with each other to produce offspring that can also reproduce. The offspring are fertile. For example, horses belong to the species Equus caballus. Members of this species can reproduce with each other. The offspring are also horses. They belong to the same species, and they can reproduce again to produce more horses. Donkeys belong to a different species, Equus asinus. Donkeys reproduce with each other to produce fertile donkeys. But donkeys can also reproduce with horses. If a male donkey reproduces with a female horse, the offspring is a mule. Mules work very well. They are usually strong, healthy organisms, but they cannot reproduce. They are infertile. Figure 1.6a. Horses and donkeys belong to different species. B. Mules are the result of reproduction between horses and donkeys. They are infertile. The binomial naming system. You have seen that the official names for the species that horses and donkeys belong to are written in a special way. This is the way that biologists name each species on Earth. These scientific names always have two words. The naming system is therefore called the binomial system. Bi means two, and nomial means to do with names. The first name in the binomial is the name of the genus that the organism belongs to. A genus is a group of species that are related to one another. If you look at figure 1.6, you can see that horses and donkeys share a lot of features. They are obviously different species, but they belong to the same genus. In the two-word name, the genus is always written with a capital letter. The second name tells us which species the organism belongs to, and this is written with a small letter. When the binomial is printed, it is in italics. You cannot do that when you are writing, but instead you can underline the binomial. Questions. 1. Yaks have the scientific name Bos grunions. Explain what this tells us about the groups into which yaks are classified. 2. A yakolo is the offspring of a yak and a buffalo. Yakolos are unable to reproduce. Explain how this suggests that yaks and buffalo belong to different species. 1.3 Keys. If you want to identify an organism whose name you do not know, you may be able to find a picture of it in a book. However, not every organism may be pictured, or your organism may not look exactly like any of the pictures. If this happens, you can use a dichotomous key to work out what your organism is. Dichotomous means branching into two. A dichotomous key is a way of leading you through to the name of your organism, by giving you two descriptions at a time and asking you to choose between them. Each choice you make then leads you on to another pair of descriptions, until you end up with the name of your organism. Here is a key that you could use to identify the organisms shown in figure 1.7. 1. Jointed limbs. 2. No jointed limbs. Earthworm, 2 more than 5 pairs of jointed limbs. Centipede 5 or fewer pairs of jointed limbs. 3. First pair of limbs form large claws. Crab, no large claws. 4. 43 pairs of limbs. Locust, 4 pairs of limbs. Spider. Figure 1.7. 5 organisms for practicing using a key. To use the key, choose one of the animals that you are going to identify. Let's say you choose organism B. Look at the first pair of statements in the key. Decide which description in step 1 matches your organism. It has jointed limbs, so the key tells us to go to step 2. Look at the descriptions in step 2. Decide which describes organism B. It has more than 5 pairs of jointed limbs, so it is a centipede. Now try working through the key to identify the other 4 animals. Notice that, in the key, each pair of statements are opposites of one another. In 1, for example, the 2 statements are about whether the organism has jointed limbs or not. Remember this when you are writing your own keys. Don't mix different ideas into a pair of statements. Activity 1.2. Constructing a key. Using a key is quite easy, but writing your own key is much more of a challenge. Work with a partner for this activity. You are going to write a key to enable someone to identify each of the 4 flowers in Figure 1.8. Figure 1.8. 4 flowers for identification using a key. First, make a list of features that clearly differ between the flowers. They should be features that cannot possibly be mistaken. Remember that the person using the key will probably only have one of the flowers to look at, so they cannot necessarily compare it with another kind of flower. Therefore, the number of petals or the color is a good choice, but the size is not, because different people might have different ideas about what is large or small. Choose one of the features that can split the flowers into 2 groups. The 2 groups don't have to be the same size. You could have 2 in one group and 2 in the other, or perhaps 1 in one group and the other 3 flowers in the second group. Write down the 2 statements for this feature. Remember that the 2 statements need to be opposites of one another. Next, concentrate on a group that contains more than 1 flower. Choose another feature that will allow you to split the flowers into 2 further groups. Keep doing this until each group contains only 1 flower. Go back to your key and make changes to improve it. Think carefully about the wording of each pair of statements. Make sure that each pair is made up of 2 clear alternatives. Try to reduce your key to the smallest possible number of statement pairs. Finally, try your key out on a friend. If they have any problems with it, then try to reword or restructure your key to make it easier to use. Self-assessment. Rate yourself according to the following scheme for each of the points listed. If you did it really well. If you made a good attempt at it and partly succeeded if you did not try to do it, or did not succeed. I had my own ideas, but I also listened to my partner's ideas. We wrote the descriptions in the key very clearly. We chose descriptions that other people could easily use. The 2 statements in each pair were opposites of one another. Our key had no more than 3 pairs of statements. When we asked other people to try our key, they found it easy to use. Reflection. Describe one thing that you think you did very well when you worked on constructing your key. Now describe one thing that you will try to do better next time you construct a key. 1.4 Kingdoms. We have seen that the species is the smallest group into which living organisms are classified. Now we will look at the largest groups. These are the kingdoms of living organisms. The kingdoms with which we are most familiar are the animal kingdom and the plant kingdom. The animal kingdom. Animals are usually easy to recognize. Most animals can move actively, looking for food. Under the microscope, we can see that their cells have no cell walls. Figure 1.9, jellyfish, birds and butterflies all belong to the animal kingdom. Characteristic features of animals. Their cells have a nucleus, but no cell walls or chloroplasts. They feed on organic substances made by other living organisms. The plant kingdom. The plants that are most familiar to us are the flowering plants, which include most kinds of trees. These plants have leaves, stems, roots and flowers. However, there are other types of plants including ferns and mosses that do not have flowers. What all of them have in common is the green color, caused by a pigment called chlorophyll. This pigment absorbs energy from sunlight, and the plant can use this energy to make sugars, by the process of photosynthesis. Figure 1.10, many kinds of plant have roots, stems and leaves. Some also have flowers. Figure 1.11, moss, saguaro cacti and coconut palms belong to the plant kingdom. Because they do not need to move around to get their food, plants are adapted to remain in one place. They often have a spreading shape, enabling them to capture as much sunlight energy as possible. Under the microscope, we can see that the cells of plants have cell walls. Characteristic features of plants. Their cells have a nucleus and cell walls made of cellulose and often contain chloroplasts. They feed by photosynthesis. They may have roots, stems and leaves. Questions. 3. Figure 1.12 shows a sea anemone, which belongs to the animal kingdom. Figure 1.12, a sea anemone. In the past, people used to think that sea anemones were plants. Suggest why. B. Explain how using a microscope could help you to confirm that sea anemones are animals. 4. Figure 1.13 shows part of a plant called a liverwort. Liverworts do not have roots or flowers. Suggest how you could show that a liverwort belongs to the plant kingdom. Figure 1.13, part of a liverwort. The 5 kingdoms. As well as the animal kingdom and the plant kingdom, there are 3 other kingdoms into which organisms are classified. These are the fungus, prokaryote and protoctist kingdoms. The fungus kingdom. For a very long time, fungi were classified as plants. However, we now know that they are really very different, and belong in their own kingdom. Figure 1.14 shows some of the features of fungi. Figure 1.14, 2 examples of fungi. Mushrooms and toadstools are fungi, and they can be colorful and easy to spot. However, the main body of most fungi, including mushrooms and toadstools, is made up of microscopic threads called hyphae. These are made of many cells joined end to end. The cells have cell walls, but these are not made of cellulose. Figure 1.15a, these toadstools are feeding on dead wood. Their hyphae grow through the wood, taking up nutrients from it. B. The mold on this strawberry is a fungus. Fungi do not have chlorophyll and do not photosynthesize. Instead they feed saprophytically, or parasitically, on organic material such as feces, human foods and dead plants or animals. Many fungi are decomposers, breaking down waste material from other organisms and dead organisms. This helps to return nutrients to the soil that other organisms can use for their growth. Fungi reproduce by forming spores. These are tiny groups of cells with a tough, protective outer covering. They can be spread by the wind or animals, and grow to form a new fungus. We have found many different uses to make of fungi. We eat them as mushrooms. We use the single-celled fungus, yeast, to make ethanol and bread. You can find out about this in Chapter 20. We obtain antibiotics such as penicillin from various different fungi. Some fungi, however, are harmful. Some of these cause food decay, while a few cause diseases, including ringworm and athlete's foot. Characteristic features of fungi. They are usually multicellular, but some such as yeast are unicellular. They have nuclei and cell walls, but the walls are not made of cellulose. They do not have chlorophyll. They feed by digesting waste organic material and absorbing it into their cells. The protoctist kingdom. The kingdom Protoctista contains a mixed collection of organisms. They all have cells with a nucleus, but some have plant-like cells with chloroplasts and cellulose cell walls, while others have animal-like cells without these features. Most protoctists are unicellular, but some, such as seaweeds, are multicellular. Figure 1.16. Two examples of protoctists. Paramecium has animal-like cells, while Chlamydomonas has plant-like cells. Figure 1.17. This photograph shows some pond water seen through a microscope. All of the organisms are protoctists. Some are unicellular, and some are multicellular. Some have cells like animal cells, while others have cells like plant cells. Characteristic features of protoctists. They are multicellular or unicellular. Their cells have a nucleus and may or may not have a cell wall and chloroplasts. Some feed by photosynthesis, and others feed on organic substances made by other organisms. The prokaryote kingdom. The prokaryote kingdom contains a huge number of organisms, but we are often completely unaware of them. Bacteria belong to this kingdom. Bacteria have cells that are very different from the cells of organisms in the other four kingdoms. The most important difference is that they do not have a nucleus. Figure 1.18. The structure of a prokaryotic cell. Figure 1.19. This photograph was taken using an electron microscope and shows bacteria that can live in the alimentary canal and can make you feel ill. It has been magnified approximately 10,000 times. You will meet bacteria at various stages in your biology course. Some of them are harmful to us and cause diseases such as cholera. Many more, however, are helpful. You will find out about their useful roles in the carbon cycle and the nitrogen cycle in Chapter 18, and their uses in biotechnology in Chapter 20. Some bacteria can carry out photosynthesis. The oldest fossils belong to this kingdom, so we think that they were among the first kinds of organism to appear on Earth. Recently, biologists have discovered bacteria living in rocks more than 3 kilometers beneath the Earth's surface. Most organisms belonging to this kingdom are unicellular, and so their characteristic features are to do with the structure of their cells. This is described in more detail in Chapter 2, where you will find explanations of some of the terms below that may be unfamiliar. Characteristic features of prokaryotes. They are usually unicellular. They have no nucleus. They have cell walls, not made of cellulose. They have no mitochondria. They have a circular loop of DNA, which is free in the cytoplasm. They often have plasmids. Questions. 5. Staphylococcus aureus is a bacterium that is often found on human skin. A. Name the genus to which this bacterium belongs. B. Name the kingdom to which this bacterium belongs. C. Describe 2 ways in which the structure of Staphylococcus aureus differs from the structure of a plant cell. 6. Figure 1.20 shows part of a fungus. The photograph was taken with an electron microscope. A. Name the structure labeled A. B. Explain how the cells in this structure differ from the cells of an organism belonging to the animal kingdom. C. The structure labeled B contains spores. What is the function of this structure? Figure 1.20, part of a fungus. Activity 1.3. Making a display about a kingdom of organisms. Work in a group of 3 or 4 for this activity. In your group, choose one of the kingdoms of organisms. Try to make sure that at least one group is working on each kingdom. Make a poster or other type of display to describe the features of this kingdom. Include photographs or drawings of several different organisms that belong to this kingdom. 1.5 Groups within the animal and plant kingdoms. Within each kingdom, biologists classify organisms into smaller groups. In the animal kingdom, these groups include vertebrates and arthropods. If you are studying the supplement, you also need to know about some of the groups in the plant kingdom. Vertebrates. Vertebrates are animals that have backbones. These are the most familiar animals: fish, amphibians, reptiles, birds and mammals. Fish. Fish all live in water, apart from a few species such as mudskippers that are able to come onto land for short periods of time. Figure 1.21a. A great white shark. B. Masked butterfly fish. Figure 1.22. A fish. Characteristic features of fish. They are vertebrates with scaly skin. They have gills throughout their life. They have fins. Their eggs have no shells and are laid in water. Amphibians. Most adult amphibians live on land. However, they always go back to the water to breed. The larvae are called tadpoles, and they spend the first part of their life in water. They then undergo a major change in the shape of their body, called metamorphosis, as they become an adult. Frogs, toads and salamanders are amphibians. Figure 1.23. Two examples of amphibians. A. A salamander. B. A toad. Figure 1.24. A frog, an example of an amphibian. Characteristic features of amphibians. They are vertebrates with skin with no scales. Their eggs have no shells and are laid in water. The tadpoles live in water, but adults often live on land. The tadpoles have gills for gas exchange, but adults have lungs. Reptiles. These are the crocodiles, lizards, snakes, turtles and tortoises. Reptiles all have scales on their skin. Unlike amphibians, reptiles do not need to go back to the water to breed because their eggs have a soft, but waterproof shell which stops them from drying out. Figure 1.25a. This Nile crocodile has just hatched from its soft-shelled egg. B. Dinosaurs, including Tyrannosaurus rex, are a group of reptiles that became extinct about 65 million years ago, but we know a lot about them from fossils. Figure 1.26. A snake, an example of a reptile. Characteristic features of reptiles. They are vertebrates with scaly skin. They lay eggs with soft shells. Birds. Birds evolved from dinosaurs, and some biologists consider that they are dinosaurs with feathers. Birds are easy to identify because they are the only animals with feathers. However, they sometimes do have scales like reptiles, but generally only on their legs. The other very distinctive feature is their beak. Figure 1.27a. Although ostriches have wings, they cannot fly. B. The oriental dwarf kingfisher lives in forests in India and Southeast Asia. Despite its name, it eats small frogs and crickets rather than fish. Figure 1.28. A bird. Characteristic features of birds. They have feathers. They have a beak. Their front two limbs are wings. They lay eggs with hard shells. Mammals. The mammals are the most familiar vertebrates, and people who have not studied biology often use the word animal to mean mammal. Humans, of course, are mammals. Some of the features of mammals are shared with birds. For example, both mammals and birds control their body temperature and have a heart with 4 chambers. Mammals also give birth to live young, but this is not helpful in identifying them, because many fish and reptiles also do this. The easiest way to identify a mammal is that its skin has hair. Looking more closely, we find that their ears have a pinna on the outside, and that they have different kinds of teeth. But it is their method of reproduction and caring for their young that is most distinctive. Only mammals have a uterus and placenta, and only mammals have mammary glands that produce milk to feed their young. Figure 1.29. 2 examples of mammals. A. A pangolin may look like a reptile at first glance, but if you look closely you can see hairs on its face and feet. B. A baby sperm whale sucks milk from its mother's mammary glands. Figure 1.30. Some of the visible external features of a mammal. Characteristic features of mammals. They have hair on their skin. Their young develop in a uterus, Attached to the mother by a placenta. The females have mammary glands, which produce milk to feed their young. They have different kinds of teeth. They have a pinna on the outside of the body. They have sweat glands in the skin. They have a diaphragm. Questions. 7. Name the kingdom to which mammals belong. 8. List 2 differences between amphibians and reptiles. 9. List a 2 external and b 2 internal features of mammals that are not found in other groups of vertebrates. Arthropods. Arthropods are animals with jointed legs, but no backbone. They are a very successful group, because they have a waterproof exoskeleton. An exoskeleton is on the outside of the body, rather than on the inside like yours. The exoskeleton supports arthropod bodies, and also allows these animals to live on land without drying out. There are more kinds of arthropod in the world than all the other kinds of animal put together. Characteristic features of all arthropods. They have several pairs of jointed legs. They have an exoskeleton. There are several different groups of arthropods, including insects, crustaceans, arachnids and myriapods. Insects. Insects are a very successful group of arthropods. Their success is mostly due to their exoskeleton and tracheae, which are very good at stopping water from evaporating from the insects' bodies, so they can live in very dry places. They are mainly terrestrial. Figure 1.31, a dragonfly, an example of an insect. Figure 1.32, features of a typical insect. Characteristic features of insects. They are arthropods with 3 pairs of jointed legs. They have 2 pairs of wings. They breathe through tubes called tracheae. Their body is divided into a head, thorax and abdomen. They have 1 pair of antennae. Crustaceans. These are the crabs, lobsters and woodlice. They breathe through gills, so most of them live in wet places and many are aquatic. Figure 1.33, a lobster, an example of a crustacean. You can clearly see the 2 pairs of antennae on its head. Figure 1.34, features of a crustacean. Characteristics. They are arthropods with more than 4 pairs of jointed legs. They have 2 pairs of antennae. Arachnids. These are the spiders, ticks and scorpions. Arachnids are generally land-dwelling organisms. Figure 1.35, a scorpion, an example of an arachnid. Figure 1.36, features of an arachnid. Characteristics. They are arthropods with 4 pairs of jointed legs. They have no antennae. Their body is divided into 2 parts, a cephalothorax and abdomen. Myriapods. These are the centipedes and millipedes. Figure 1.37, a giant African millipede, an example of a myriapod. Figure 1.38, features of a myriapod. Characteristics. Their body consists of many similar segments. Each of their body segments has jointed legs. They have 1 pair of antennae. Questions. 10. Alice 2 features that are shared by myriapods and arachnids. B. Describe 2 differences between myriapods and arachnids. 11. Fleas are insects, but they do not have wings. Suggest 2 features of fleas that would show that they should be classified as insects. Activity 1.4. Classifying animals. In this activity, you practice identifying the classification groups to which animals belong. You will need pictures or specimens of different vertebrates and arthropods, arranged around the room. A clipboard. Work with a partner for this activity. First, read ahead to find out what you are going to do. On a sheet of paper, draw a table in which you can fill in your answers. Look carefully at the first specimen. Discuss with your partner whether it is a vertebrate or an arthropod, and then decide which smaller group it belongs to within these two large groups. Write down your decision, and the reasons for it. Repeat for as many of the other specimens as you have time for. Reflection. How are you going to try to remember the features of the four groups of arthropods? Which of these ideas, or a combination of them, do you think might work best for you? Looking at labeled diagrams and reading lists of features. Trying to write your own list of features. Writing features on cards and then sorting them to match each group. Testing a friend or asking them to test you. Ferns. Ferns are plants with leaves called fronds. They do not produce flowers, but instead reproduce by means of spores produced on the underside of the fronds. Most ferns are quite small, but some species can be as much as 20 m tall. Figure 1.39, Tree ferns in Bali, Indonesia. Figure 1.40, Features of a fern. Characteristic features of ferns. They are plants with roots, stems and leaves. They do not produce flowers. They reproduce by spores produced on the undersides of their fronds. Flowering plants. These are the plants that are most familiar to us. As their name suggests, they reproduce by producing flowers. You will find out about their transport systems and method of reproduction in chapters 8 and 14. Figure 1.41, Two examples of flowering plants. A. An iris, an example of a monocot plant. B. Hibiscus, an example of a dicot plant. Characteristic features of flowering plants. They are plants with roots, stems and leaves. They reproduce using flowers and seeds. Their seeds are produced inside an ovary, in the flower. Flowering plants can be divided into two main groups: dicotyledons and monocotyledons. These names refer to the structure of their seeds. The seeds of flowering plants contain seed leaves or cotyledons. Monocotyledons have only one cotyledon in their seeds, whereas dicotyledons have two. Figure 1.42, Features of a monocot plant. Figure 1.43, Features of a dicot plant. Characteristic features of dicots. They have seeds with two cotyledons. They usually have a main root with side roots coming out from it. Their leaves have a network of veins. They have flower parts in multiples of 4 or 5. They have vascular bundles in the stem, arranged in a ring. Characteristic features of monocots. They have seeds with one cotyledon. Their roots grow out directly from the stem. Their leaves have parallel veins. They have flower parts in multiples of 3. They have vascular bundles in the stem, arranged randomly. Questions. 12. Describe two differences between ferns and flowering plants. 13. Inches your local area. Find two examples of monocotyledons and two examples of dicotyledons. Explain how you were able to decide which group each plant belonged to. Activity 1.5. Classifying animals and plants. In this activity, you practice identifying the classification groups to which organisms belong. You will need pictures or specimens of organisms belonging to the animal, plant, fungus, prokaryote and protoctus kingdoms, arranged around the room. Pictures of ferns and flowering plants, some dicotyledons, and some monocotyledons, also arranged around the room. A clipboard. Work with a partner for this activity. First, read ahead to find out what you are going to do. On a sheet of paper, draw a table in which you can fill in your answers. Look carefully at the first specimen. Discuss with your partner which group it belongs to. Write down your decision and the reasons for it. Repeat for as many of the other specimens as you have time for. 1.6 Viruses. You have almost certainly had an illness caused by a virus. Viruses cause common diseases such as colds and influenza, and also more serious ones such as AIDS. Viruses are not normally considered to be living organisms, because they cannot do anything other than just exist until they get inside a living cell. They then take over the cell's machinery to make multiple copies of themselves. These new viruses burst out of the cell and invade others, where the process is repeated. The host cell is usually killed when this happens. On their own, viruses cannot move, feed, excrete, show sensitivity, grow or reproduce. They do not display the 7 characteristics of living things. Figure 1.44 shows one kind of virus. It is not made of a cell, it is simply a piece of genetic material surrounded by a protein coat. It is hugely magnified in this diagram. The scale bar represents a length of 10 nanometers. 1 nanometer is 1/10 m. In other words, you could line up more than 15,000 of these viruses between 2 of the millimeter marks on your ruler. Figure 1.44, the structure of a virus. Question. 14. Figure 1.45 shows a virus. With reference to the diagram, and your own knowledge, discuss whether or not viruses can be considered to be living organisms. Figure 1.45, a virus. Summary. All organisms show 7 characteristics: movement, respiration, sensitivity, growth, reproduction, excretion and nutrition. Organisms are classified into groups according to features that they share. Classification systems reflect evolutionary relationships between organisms. The binomial system of naming organisms shows the genus and species. The name of the genus has a capital letter, followed by the name of the species with a small letter. Dichotomous keys are used to identify organisms. They are made up of pairs of contrasting statements or questions. Taking one organism at a time, you work through the key until it leads you to the name of the organism. When constructing a dichotomous key, it is important to make sure that a person using the key can make a decision looking at only one organism. Comparative or subjective descriptions are not suitable. Organisms belonging to the plant kingdom have cells with cell walls made of cellulose, and some of their cells have chloroplasts. Organisms belonging to the animal kingdom have cells that do not have these features. Vertebrates are animals with backbones. They are classified in 5 main groups: fish, amphibians, reptiles, birds and mammals, each with their own set of distinguishing features. Arthropods are animals with an exoskeleton and jointed legs. They include insects, arachnids, crustaceans and myriapods, which can be distinguished from each other by the number of legs and antennae. As well as the animal and plant kingdoms, organisms are classified into the fungus, prokaryote and protoptous kingdoms. They differ in the structure of their cells. Ferns and flowering plants are 2 groups within the plant kingdom. Flowering plants can be classified as monocots or dicots, which differ in the patterns of veins in their leaves, the structure of their root systems, the number of flower parts, and the distribution of vascular bundles in their stems. Viruses are not classified in any of the 5 kingdoms, as they do not show the characteristics of living organisms. They do not have cells, and consist of genetic material surrounded by a protein coat. Project. A new species. Each year, biologists discover new species. Some of these are small while others are surprisingly large. Work in a group of 3 or 4. Use the internet to search for some examples of newly discovered species, and select one to research in detail. Decide how you will share the results of your research with others. For example, you could give an illustrated talk, or produce a poster. Decide how you will share out the tasks between you. Try to find information about some or all of these issues. Where and how was the new species discovered? Why had it not been discovered before? How did biologists decide that it really was a new species? How was the binomial for the new species chosen? What does its name mean? Biologists will want to find out more about the new species, but if it is rare they will not want to take many specimens from the wild, or disturb it in its habitat. How have these conflicts been resolved? Figure 1.46. The tree Dinesia juaranificau is a species that was recently discovered in Brazil. Exam-style questions tip. The mark allocation for a question will often indicate the number of points that you should make in order to fully answer that question. 1. Which characteristic is not shown by all living organisms? E. Excretion, movement, photosynthesis. Respiration. 2. Which feature is found in all vertebrates and all arthropods? A. Backbone, band, exoskeleton, cantennae. D. Cells without cell walls. 3. The binomial of the okapi is Okapia johnstoni. What genus does the okapi belong to? Animals beyond stonic mammals. D. Okapia walls. 4. Which are features of monocotyledons? A phloem parts in multiples of 4 or 5, 1 cotyledon in seeds, bone cotyledon in seeds, vascular bundles in a ring, vascular bundles in a ring, network of veins in leaf. D. Parallel veins in leaf, flower parts in multiples of 3. 5. What 2 features do all viruses possess? A cell membrane, cell wall. Vitoplasm, nucleus. Genetic material, protein coat. Ribosomes, plasmid 6. Diagram above shows 5 leaves. Use the key to identify the species of plant that each of these leaves came from. 1. Veins are parallel. Go to 2. Veins are branching. Go to 3. 2. The leaf is more than 5 times as long as it is wide. Iris germanica. The leaf is less than 5 times as long as it is wide. Tricertus hirta. 3. Leaf has a prickly edge. Ilex. Aquifolium. Leaf does not have a prickly edge. Go to 4. 4. Leaf is divided into many leaflets. Fraxinus excelsior. Leaf is not divided into many leaflets. Buddleia davidii. By explain the meaning of the term species. 2 biologists give each species a 2-word Latin name. What is the term used to describe this naming system? 7. The table shows features of 3 animals. 3 species of tree have the following Latin names. Carpodiptera africana. Comifera africana. Comifera angolensis. Which 2 species do biologists consider to be the most closely related? Explain your answer. B. Comifera africana is a dicotyledon. State 2 features that it shares with all other dicotyledons, but not with monocotyledons. 9. All living organisms are classified into 5 kingdoms. These include the plant, animal and fungus kingdoms. A. Name the other 2 kingdoms. B. Describe 2 ways in which the cells of fungi differ from those of plants. See the diagram below shows a virus. I. Name parts 10 and why. 2. Explain why viruses are not generally classified into any of the 5 kingdoms. Self-evaluation checklist. After studying this chapter, think about how confident you are with the different topics. This will help you to see any gaps in your knowledge and help you to learn more effectively. Chapter 2. Cells. In this chapter you will find out about the structure of the cells of animals, plants and bacteria. Learn about the functions of each of the parts of these cells. Describe how the structures of some specialized cells are related to their functions. Practice using the magnification equation. Getting started. Organisms that belong to different kingdoms have different kinds of cells. With a partner, think about the answers to these questions. 1. If you have a microscope, how can you distinguish between a cell of an organism belonging to the plant kingdom and one belonging to the animal kingdom? 2. Still using your microscope, how could you tell if a cell belongs to an organism from the prokaryote kingdom? Cells from deep time. Despite its inviting blue color, you would not want to get into the water in this hot spring. The temperature of the water can be as high as 90 degrees Celsius in Yellowstone National Park, USA. But there are organisms that can live in this hot water. They are called extremophiles, meaning organisms that like extreme conditions. They produce the colors around the edge of the pool. Many of these organisms are bacteria. Bacteria are made of single cells. Their cells are much less complex than animal or plant cells. For example, they do not have a nucleus. Scientists think that bacteria like these first evolved more than 3.5 billion years ago, when conditions on Earth were quite similar to conditions in some hot springs today. This was long before animals and plants first appeared on Earth. Despite their simple structure, bacteria are clearly very successful. They have survived on Earth almost unchanged, through an unimaginably long period of time. Discussion questions. 1. What evidence might lead scientists to think that bacteria evolved more than 3.5 billion years ago? 2. We tend to think of complex organisms such as humans as being more advanced than simple ones such as bacteria. But bacteria have lived on Earth for 3.5 billion years, and are still here and doing well. Do you think that humans really are more advanced than bacteria? 2.1 Animal and plant cells. All organisms are made of cells. Cells are very small, so large organisms contain millions of cells. They are multicellular. Some organisms are unicellular, which means that they are made of just a single cell. Bacteria and yeast are examples of single-celled organisms. All cells are made from existing cells. New cells are formed when a fully grown cell divides. You can read about how cells divide in Chapter 16. Microscopes. To see cells clearly, you need to use a microscope. The kind of microscope used in a school laboratory is called a light microscope. This is because it shines light through the piece of animal or plant you are looking at. It uses glass lenses to magnify and focus the image. A very good light microscope can magnify about 1,500 times, so that all the structures in figures 2.2 and 2.3 can be seen. Figure 2.1, a light microscope. Figure 2.2, an animal cell as it appears through a light microscope. Figure 2.3, a plant cell as it appears through a light microscope. A photograph taken using a light microscope is called a photomicrograph. Figure 2.4 is a photomicrograph of some animal cells, and Figure 2.5 is a photomicrograph of some plant cells. Figure 2.4, these are cells from the trachea of a mammal. They have been stained with a dye that makes the nucleus look darker than the cytoplasm. Figure 2.5, these are cells from a moss plant. You cannot see the cell membranes because they are pressed tightly against the inside of the cell walls. To see smaller things inside a cell, an electron microscope is used. A picture taken with an electron microscope is called an electron micrograph. This type of microscope uses a beam of electrons instead of light, and can magnify things up to 500,000 times. This means that a lot more detail can be seen inside a cell. We can see many structures more clearly, and can see some structures that cannot be seen at all with a light microscope. Figure 2.6. These are cells from the leaf of a maize plant, seen using an electron microscope. Electron microscopes do not provide images that show the colors of the different parts. Often, color is added to the image afterwards. Questions. 1. Explain the difference between a unicellular organism and a multicellular organism, giving one example of each. 2. Look at the micrographs in figures 2.4 and 2.5. Suggest why a stain has been added to the animal cells, but we can easily see the plant cells without using a stain. Cell membrane. Every cell has a cell membrane around the outside. The cell membrane is a very thin layer of protein and fat. It is very important to the cell because it controls what goes in and out of it. It is said to be partially permeable, which means that it will let some substances through but not others. The cell membrane separates the contents of the cell from its environment. It is difficult to see the cell membrane in a plant cell because it is pressed tightly against the inside of the cell wall. Cell wall. All plant cells are surrounded by a cell wall made mainly of cellulose. Paper, which is made from cell walls, is also made of cellulose. Animal cells never have cell walls. Cellulose belongs to a group of substances called polysaccharides, which are described in Chapter 4. Cellulose forms fibers, which crisscross over one another to form a very strong covering to the cell. This helps to protect and support the cell. If the cell absorbs a lot of water and swells, the cell wall stops it bursting. Figure 2.7. This photograph of a plant cell wall was taken with a kind of electron microscope that shows surfaces in 3 dimensions. You can see how the fibers of cellulose crisscross over one another. Because of the spaces between fibers, even very large molecules are able to go through the cellulose cell wall. It is described as fully permeable. Cytoplasm. Cytoplasm is a clear jelly. It is nearly all water. About 70% is water in many cells. It contains many substances dissolved in it, especially proteins. Many different metabolic reactions take place in the cytoplasm. Vacuole. A vacuole is a fluid-filled space inside a cell which is surrounded by its own membrane. Plant cells have very large, permanent vacuoles, which contain a solution of sugars and other substances called cell sap. When the vacuole is full, it presses outwards on the rest of the cell and helps it to keep its shape. Animal cells have much smaller vacuoles, called vesicle, which also contain solutions. Nucleus. The nucleus is where the genetic information is stored. The information is kept on the chromosome, which are inherited from the organism's parents. The chromosomes are made of DNA. The information carried on the DNA determines the kinds of proteins that are made in the cell. You can find much more about this in Chapter 16. Chromosomes are very long, but so thin that they cannot easily be seen even using an electron microscope. However, when the cell is dividing, they become short and thick, and can be seen with a good light microscope. Figure 2.8. This photomicrograph shows some plant cells dividing. The nuclei of the cells have been colored with a blue stain. The things that look like spider legs are chromosomes. We can normally only see chromosomes when a cell is dividing. Chloroplasts. Chloroplasts are never found in animal cells, but most of the cells in the green parts of plants have them. They contain the green coloring or pigment called chlorophyll. Chlorophyll absorbs energy from sunlight, and this energy is then used in making food for the plant by photosynthesis. Chloroplasts often contain starch grains, which have been made by photosynthesis. Animal cells never contain starch grains. Figure 2.9. This electron micrograph shows a chloroplast from a cell in a pea plant. The large blue structure inside the chloroplast is a starch grain. The black stripes inside the chloroplast are membranes where the chlorophyll is kept. Mitochondria. All the structures we have looked at so far, cell membrane, cell wall, cytoplasm, vacuoles, nucleus and chloroplasts, can be seen with a good light microscope. But there are some smaller structures inside cells that we cannot see clearly unless we use an electron microscope. These include mitochondria. Mitochondria are found in almost all plant and animal cells. Figure 2.10 is an electron micrograph of a mitochondrion, and Figure 2.11 shows what one might look like if we could cut it open and see it in 3 dimensions. Figure 2.10, an electron micrograph of a mitochondrion. The pink lines are membranes, which separate the inside of the mitochondrion from the rest of the cell. Figure 2.11, a drawing of a mitochondrion, cut open to show the membrane inside it. Mitochondria are the parts of the cell where aerobic respiration happens. This is how energy is released from glucose. You can read about aerobic respiration in Chapter 11. Aerobic respiration is the main way in which cells get the energy that they need to stay alive. The more energy a cell needs, the more mitochondria it has. Muscle cells, for example, are packed full of mitochondria. Ribosomes. Ribosomes are tiny structures found in almost all animal cells and plant cells. They are so small that we can only see them with an electron microscope. Ribosomes are where the cell makes proteins. The instructions on the DNA molecules are used to link together long chains of amino acids in a particular sequence. You can read more about protein molecules in Chapter 4, and more about how a cell makes them in Chapter 16. Figure 2.12, this diagram shows what an animal cell might look like if we cut it open. Figure 2.13, an electron micrograph showing the central part of an animal cell. Questions. 3. Name the part of a cell which has each of these functions, making proteins. B. Containing the information about which proteins to make photosynthesis. D. Preventing the cell bursting when it takes up water estering a solution of sugars and other solutes. F. Controlling what enters and leaves the cell. For which of the structures that you have named in question 3 are found in both animal and plant cells? Activity 2.1. Comparing animal cells and plant cells. Work in a group of 3 or 4 for this activity. You are going to make a display to compare the structures of animal cells and plant cells. Decide how you will do this. You could perhaps use annotated drawings, construct a large comparison table, or make a presentation. You can use the information in this chapter to make your comparison. You might also like to look for some more pictures on the internet. Activity 2.2. Functions of the parts of a plant cell. You could work on your own or in a pair for this activity. You will need a large sheet of paper, at least A4 and larger if possible. A pencil, marker pens and a ruler. On your large sheet of paper, make a drawing of a plant cell. You could copy the one in Figure 2.3. Leave plenty of space around your drawing. 2. Use the information in this chapter to find the function of one part of the cell, for example, the cell wall. 3. Draw a label line, using a ruler, to this part of the cell. Write the name of the part, and then a short summary of what it does. 4. Repeat steps 2 and 3 for each part of the cell. Peer assessment. Exchange your diagram with a partner. Look at your partner's diagram and assess how well they have carried out the task. Rate them a mark according to the following scheme. Green. If they did it really well. Amber. If they made a good attempt at it and partly succeeded. Red. If they did not try to do it, or did not succeed. You could think about whether the drawing is as large and clear as possible. How well they have described the function of each part. Reflection. Now that you have studied your partner's diagram, is there anything you would like to change on yours? 2.2 Bacterial cells. Bacteria are unicellular organisms. Bacterial cells are rather different from the cells of animals and plants. Figure 2.14 is a diagram of a bacterium. Figure 2.14, a bacterial cell. Bacterial cells always have a cell wall. Unlike plant cells, this cell wall is not made of cellulose. But the function is the same as in plant cells. The bacterial cell wall helps to support the cell, and stops it bursting if the cell takes up a lot of water. A partially permeable cell membrane is pressed tightly against the inside of the bacterial cell wall. As in plant and animal cells, the cell membrane controls what enters and leaves the cell. Bacterial cells have cytoplasm and ribosomes, which have the same functions as in animal and plant cells. They do not have mitochondria or chloroplasts. The most important difference between a bacterial cell and animal or plant cells is that bacteria do not have a nucleus. Bacterial cells are also known as prokaryotic cells. Pro means before, and karyotic means nucleus. Prokaryotic cells appeared on Earth millions of years before cells with nuclei appeared. Instead of chromosomes inside a nucleus, bacteria have a circle of DNA. This is sometimes called a bacterial chromosome. The DNA has exactly the same function as in other cells. It provides instructions for making proteins. Bacterial cells often have one or more smaller circles of DNA, called plasmids. Scientists can use plasmids in the genetic modification of cells and organisms, which you can read about in Chapter 20. Figure 2.15. This picture was taken with an electron microscope, after which the colors were added to it. It shows a bacterium called Enterococcus faecalis dividing into two. This is the way that bacteria reproduce. Question 5. Construct a table to compare the structure of a bacterial cell with animal and plant cells. Remember to include similarities, as well as differences. Reflection. How will you try to learn the names of the parts of animal, plant and bacterial cells, and their functions? Think about which of these ideas might work for you. Looking at diagrams and reading about the structures. Practicing drawing your own diagrams and labeling them. Getting a friend to test you by asking questions. Making some revision cards for yourself, with the name of a structure on one side and its function on the other side. What other ideas might you try? 2.3. Specialized cells. Multicellular organisms, such as humans or plants, may contain many millions of cells. Not all of these cells are alike. For example, in a human body almost all of our cells have the same features that are found in most animal cells: a cell membrane, cytoplasm, mitochondria, ribosomes, and a nucleus. But most of our cells have a particular function to perform, and their structure is modified to help them to carry out that function effectively. They are said to be specialized. The same is true in a flowering plant, where all the cells have the basic characteristics of plant cells, but then have slightly different structures that relate to their specific functions. You will meet many examples of specialized cells as you continue through your biology course. Table 2.1 lists 7 of these and indicates where you can find out more about each one. Tissues. Often, cells that specialize in the same activity are found together. A group of cells like this is called a tissue. An example of a tissue is a layer of cells lining your stomach. These cells make enzymes to help to digest your food. Figure 2.16. Cells lining the stomach, an example of a tissue. The stomach also contains other tissues. For example, there is a layer of muscle in the stomach wall, made of cells which can move. This muscle tissue makes the wall of the stomach move in and out, churning the food and mixing it up with enzymes. Plants also have tissues. You may already have looked at some epidermis tissue from an onion bulb. Inside a leaf, a layer of cells makes up the palisade tissue. These cells are specialized to carry out photosynthesis. Specialized cell. Where it is found. Function. Where you can find out more. Ciliate cell. Lining the trachea and bronchi of animals. Cilia move mucus upwards. Topic 11.2. Neuron. In the nervous system of animals. Conducting electrical impulses. Topic 12.1. Table 2.1. Examples of specialized cells. Organs. A group of different tissues that carry out a function together is called an organ. The stomach is an organ. Other organs include the heart, the kidneys and the lungs. In a plant, an onion bulb is an organ. A leaf is another example of a plant organ. The stomach is only one of the organs which help in the digestion of food. The mouth, the intestines and the stomach are all part of an organ system called the digestive system. The heart is part of the circulatory system, while each kidney is part of the excretory system. The way in which organisms are built up can be summarized like this: cells make up tissues, which make up organs, which make up organ systems, which make up organisms. 2.4 Sizes of specimens. Many of the structures that biologists study are very small. Cells, for example, are so small that we cannot see them without a microscope. The photographs and diagrams of cells in this chapter are all much larger than actual cells. We can tell someone how much bigger the image is than the actual object by giving its magnification. The magnification of an object is how many times larger it is than the real object. Magnification equals size of image size of actual object. For example, try measuring the length of the spider's body in Figure 2.17. Figure 2.17, a spider. You should find that it is 40 millimeters long. The real spider was 8 millimeters long. So, we can work out how much the diagram is magnified. Magnification equals length of body in the drawing. Length of body of the real spider equals 408 equals 5. There are two very important things to remember when you are calculating a magnification. Make sure that all the numbers in your calculation have the same units. It is often a good idea to convert everything to millimeters, millimeter, before you do anything else. Magnification is always written with a multiplication sign in front of it. Magnification does not have units. Some of the objects that we study in biology are so small that even millimeters are not a suitable unit to use for measuring them. Instead, we use micrometers. The symbol for a micrometer is m. 1 m equals 106 m, 1 m equals 106 m. Questions. 6. How many micrometers are there in 1 millimeter? 7. The mitochondrion in Figure 2.10 is magnified 20,000 times. A. Using a ruler, carefully measure the maximum length of the mitochondrion. Record your measurement in millimeter. B. Convert your answer to m. C. Use this formula to calculate the real size of the mitochondrion in m. Real size in m equals size of image in m. Magnification. How many of these mitochondria could you line up end to end between two of the millimeter marks on your ruler? Activity 2.3. Practicing using the magnification equation. Work on your own for the first part of this activity, and then pair up with someone else for the second part. You will need several small objects that are easy to measure and draw, for example an eraser, a paper clip, a button. A ruler that can measure in millimeter. Some plain paper, a good pencil and an eraser. First part, work on your own for this. 1. Measure each object carefully and write down the measurements. Don't let anyone else see these measurements at this point. 2. Make a magnified drawing of your objects. Calculate the magnification of each one and write it next to your drawings. Try to use different magnifications for each object. Second part, work with a partner. 3. Exchange your drawings with a partner. Each of you uses the drawings and magnifications to work out the size of the actual object. 4. Check your answers with your partner. Did you correctly calculate the actual sizes of the objects they had drawn? Did your partner calculate the actual sizes of the objects that you had drawn? Self-assessment. Did you calculate the magnifications of your drawings correctly, so that your partner could work out the actual size of each drawing? If not, where did you go wrong? Were you able to calculate the actual size of the objects your partner had drawn? If not, where did you go wrong? Reflection. What are the most important things that you need to remember when you use the magnification equation? What can you do to help you to remember the equation and how to use it? Summary. All organisms are made of cells. New cells are always made by the division of existing cells. Animal and plant cells contain cytoplasm, a cell membrane, ribosomes, mitochondria, and a nucleus containing DNA in the form of chromosomes. Plant cells also contain a cell wall, a large vacuole, and sometimes chloroplasts. Bacterial cells have cytoplasm, a cell membrane, a cell wall, and ribosomes. They do not have a nucleus. Their DNA is circular. They may have extra, small circles of DNA called plasmids. The cell membrane of all cells is partially permeable and controls what enters and leaves the cell. The cell wall is fully permeable and allows all molecules and ions to pass through it. Ribosomes are the site of protein synthesis in a cell. Mitochondria release energy from glucose and other nutrients by aerobic respiration. Chloroplasts are the site of photosynthesis. Cells may be specialized for specific functions. Magnification can be calculated using the equation. Magnification equals size of image size of actual object. Project. Making a model cell. It is best to work in a group of 3 or 4 for this project. Discuss different ideas about how you can make a model cell. What kind of cell will it be? An animal cell, a plant cell, or a bacterial cell? How can you construct your cell? What materials will you need? If you are short of ideas, you could search on the internet. However, even if you do find some good suggestions there, it is important to make the model cell your own, using some of your own ideas that make it different from everyone else's models. As you build your model, you will almost certainly make some changes to your original plans, so be prepared to source some extra materials as you work. After you have completed your model, you can compare it with the models that other groups have made. This might give you ideas about how you could make an even better model if you did it again. Exam-style questions. 1. The diagram below shows a cell from a plant leaf. Which labeled part contains DNA? 2. Which structure is not found in both animal and bacterial cells? A cell membrane, cytoplasm, mitochondrion. Ribosome. 3. A diagram of a flower is 8 cm across. The magnification of the diagram is 4. What is the actual width of the flower? A 3.2 mm b 20 mm c 0.5 cm. D 32 cm. 4. This diagram shows a group of palisade cells from a plant leaf. The width of the diagram is 60 mm. The mean width of an actual palisade cell is 40 m. What is the magnification of the diagram? A 1.5 b 500 c 667. D 50. 5. This is a photograph taken through a light microscope. It shows a group of plant cells. A. I state one way you can tell that these are plant cells and not animal cells. 2. State one way you can tell that these are not bacterial cells. 3. Name one cell structure that these cells do not contain, which you would expect to find in a palisade cell. B. Outline the function of each of these parts of a plant cell. I. Nucleus. I. Ribosomes. 6. The photograph shows a jellyfish. A jellyfish belongs to the animal kingdom. Actual jellyfish is 50 mm in diameter. Calculate the magnification of the photograph. B. Name 3 parts that you would find in a cell in a jellyfish and outline the function of each part. C. Some of the cells in the jellyfish are neurons. State the function of a neuron. 7. This is an electron micrograph of a small part of a cell from the pancreas. This cell makes large quantities of protein molecules, which are stored in vesicles before being exported from the cell. A. Structure A contains molecules that determine the type of proteins made in the cell. Identify structure A. Use the information above to explain why the cell has large numbers of structures B and C. Self-evaluation checklist. After studying this chapter, think about how confident you are with the different topics. This will help you to see any gaps in your knowledge and help you to learn more effectively. I can. See topic. Needs more work. Almost there. Confident to move on. Describe and compare the structure of an animal cell with a plant cell. 2.1 Describe the functions of each of the structures found in an animal cell and a plant cell. 2.1 Identify the parts of cells in diagrams and in images taken using a light microscope or an electron microscope. 2.1 Describe the structure of a bacterial cell and the functions of each of its parts. 2.2 Name some specialized cells and their functions. 2.3 Remember the magnification equation and use it to calculate magnifications or actual sizes of objects shown in images. 2.4 Convert measurements between millimeters and micrometers. 2.4 Chapter 3 Movement into and out of cells. In this chapter you will learn about diffusion and its importance to organisms. Investigate factors that affect the rate of diffusion. Find out how osmosis is a special kind of diffusion involving water. Investigate osmosis using dialysis tubing and plant tissues. Learn about active transport. Learn about water potential. Explain how osmosis affects plant cells. Explain how active transport happens. Getting started. The particle theory helps us to explain how matter behaves. Here are 6 statements about particles. Some of them describe solids, some describe liquids, and some describe gases. Which are which? The particles are far apart and rarely collide with each other. The particles vibrate on the spot. The particles are close to one another but can move around. The particles move freely in all directions. The particles stay in contact and slide past one another as they move. The particles are in fixed positions, close to one another. Using diffusion to clean the blood. We each have 2 kidneys, whose function is to remove unwanted substances from the blood. The kidneys produce a solution of the unwanted substances, which is removed from the body as urine. However, sometimes kidneys stop working. When this happens, the unwanted substances build up in the blood. Some of them can be quite dangerous, so it is important to find another way to remove them. The boy in figure 3.1 is attached to a dialysis machine. The blood from a vein in his arm flows into the machine. Inside the machine, a liquid called dialysis fluid flows past his blood. There is a very thin membrane between his blood and the dialysis fluid. The unwanted substances in his blood diffuse through the membrane into the dialysis fluid, which then flows away. His cleaned blood flows back into his body. It takes several hours for all the unwanted substances to be removed from his blood, and he has to do this several times a week. Figure 3.1, a patient undergoing dialysis. Discussion questions. 1. If the boy could have a kidney transplant, he would not have to spend time attached to a dialysis machine. Why do you think not everyone who needs a kidney transplant can have one? 2. The dialysis fluid flows constantly through the machine. Can you suggest why this works better than just having stationary fluid in the machine? 3.1 Diffusion. Everything including living cells is made of atoms, molecules and ions. These particles are always moving. The higher the temperature, the faster they move. This is because the particles have more kinetic energy at higher temperatures. The more kinetic energy they have, the faster they move. In a solid substance, the particles cannot move very far, because they are held together by attractive forces between them. They simply vibrate around a fixed position. In a liquid they can move more freely but stay in contact with one another. They bump into one another and rebound. In a gas they are freer still, with no attractive forces between the molecules or atoms. When molecules and ions are in a solution, they can move as freely as when they are liquid. Diffusion down a concentration gradient. When they can move freely, particles tend to spread themselves out as evenly as they can. This happens with gases, solutions, and mixtures of liquids. Imagine, for example, a rotten egg full of hydrogen sulfide gas in one corner of a room. Hydrogen sulfide gas is very smelly. To begin with, there is a high concentration of the gas near the egg, but none in the rest of the room. The hydrogen sulfide molecules quickly spread through the air in the whole room. Soon, you cannot tell where the smell first came from. The whole room smells of rotten egg. Figure 3.2. The random movements of gas molecules result in them spreading evenly through all the space available. The hydrogen sulfide molecules have spread out through the air. This spreading out is called diffusion. Diffusion happens because the molecules move randomly. They do not purposefully move from one place to another. Each molecule just moves by chance, changing direction when it bumps into another molecule. Some molecules might even go back into the rotten egg again. But overall, the molecules spread fairly evenly across the room. We call this the net movement of the molecules. Some go from the egg into the room, and a few from the room back into the egg, but overall, most of them go from the egg into the room. A place where a lot of the molecules are quite close together has a high concentration of molecules. In this example, the rotten egg had the highest concentration of hydrogen sulfide molecules. The rest of the room had a low concentration of hydrogen sulfide molecules. Overall, the random movements of the molecules caused them to spread out evenly throughout all of the space they could get to. One way of thinking about this is to imagine a slope. The place with the high concentration is at the top of the slope. The place with the low concentration is at the bottom of the slope. The molecules move down the slope from where there is a high concentration to where there is a low concentration. Another word for slope is gradient. We can therefore say that there is a concentration gradient from the rotten egg to the rest of the room. The overall result of diffusion is that the hydrogen sulfide molecules move down this concentration gradient. Activity 3.1. Modeling diffusion. In this activity, you will model diffusion using people to represent particles. You will need a fairly big, empty space to move around in, e.g. a playground or a large hall. Work as a whole class for this activity. Each person in the class represents a particle in a gas. Start with most people in one corner of the room or space. Don't crowd too closely. Remember that gas particles do not touch one another. Face any way that you like. Each person now starts to move in a straight line in the direction they are facing. Keep going in your straight line until you hit something. This might be another particle, or it might be the wall of your container. When you collide, change direction and keep going in your new straight line. Continue doing this for a few minutes. Then stop and look around you. Questions. 1. What has happened to the particles? Are they still concentrated in one corner? 2. Try to explain why this happened. Reflection. You have now used two different models of diffusion. The first one was the picture of a slope in Figure 3.3, and the second model used moving people to represent particles. Figure 3.3. This diagram shows how we can use the analogy of a slope to think about a concentration gradient. We can imagine particles moving down the slope, from a high concentration to a low concentration. Do either of these models help you to understand diffusion? Why do you think that is? Diffusion in living organisms. Living organisms get many of the substances they need by diffusion. They also get rid of many of their waste products in this way. For example, plants need carbon dioxide for photosynthesis. This diffuses from the air into the leaves, through the stomata. It does this because there is a lower concentration of carbon dioxide inside the leaf, as the cells are using it up. Outside the leaf in the air, there is a higher concentration. Carbon dioxide molecules therefore diffuse into the leaf, as a result of their random movement, down this concentration gradient. Oxygen, which is a waste product of photosynthesis, diffuses out in the same way. There is a higher concentration of oxygen inside the leaf, because it is being made there. Oxygen therefore diffuses out through the stomata into the air. Diffusion is also important in gas exchange for respiration in animals and plants. Cell membranes are freely permeable to oxygen and carbon dioxide, so these gases easily diffuse into and out of cells. Figure 3.4. Some substances, such as oxygen and carbon dioxide, move into and out of cells by diffusion through the cell membrane. Oxygen and carbon dioxide are both gases, but living organisms also rely on diffusion of solutes. A solute is a substance that has dissolved in a liquid, which is called a solvent. In organisms, the solvent is always water. For example, the cytoplasm of all cells contains many solutes, such as glucose molecules or sodium ions. These particles are free to move through the cytoplasm, so they diffuse, spreading to all parts of the cytoplasm. Questions. 1. Look at the definition of diffusion in the keywords box. Explain what each of these words in the definition means. A net movement B concentration gradient random movement. 2. When substances diffuse into and out of cells, which part of the cell must they move through? 3. Describe 3 examples of diffusion in organisms. For each example, state whether the substance that is diffusing is a gas or a solute. 4. Where does the energy for diffusion come from? Experimental skills 3.1. Investigating how surface area affects diffusion. In this investigation, you will use pieces of agar jelly to represent cells. You will follow instructions to investigate how the surface area of the cells affects the diffusion of a substance through them. You will need a dish containing agar jelly, made with a little bit of a weak alkali such as ammonium hydroxide, and colored with a dye called cresol red. A sharp knife to cut the jelly into pieces. Some plastic or blunt-ended forceps. A ruler to measure in millimeter. 2 large test tubes or other large glass tubes, more than 1 centimeter in diameter. A stopwatch. Some dilute hydrochloric acid. Safety, take care with the acid. Wear safety glasses in case of splashes. If you spill it, clean up the spills immediately, using plenty of cold water. Take care with the sharp blade. Cut with the blade facing away from you. Place the jelly on a firm surface before you start to cut it. Method. 1. Cut 2 cubes of agar jelly, with sides of 1 centimeter. 2. Cut one of the cubes into 4 smaller cubes, each with sides of 0.5 centimeters. 3. Put the large cube into a test tube. Put the 4 small cubes into the other test tube. 4. Add equal volumes of dilute hydrochloric acid to each tube, making sure that all of the cubes are covered with acid. 5. Start the stopwatch. Time how many seconds it takes for the cubes in each test tube to become fully yellow. Questions. 1. The jelly cubes contained a weak alkali and cresol red, which is an indicator. Explain why the cubes changed color. 2. How did the volume of the 4 small cubes compare with the volume of the 1 larger cube? Was it greater, smaller, or the same? 3. How did the total surface area of the 4 small cubes compare with the surface area of the 1 larger cube? Was it greater, smaller, or the same? 4. Copy and complete these sentences, choosing the correct words. The time taken for the 4 small cubes to completely change color was greater smaller than the 1 single cube. This is because the surface area volume of the small cubes was greater smaller than for the single cube. 5. Write a conclusion for your experiment. Include the words diffusion and surface area in your conclusion. Experimental Skills 3.2. Investigating how temperature, concentration gradient, or distance affect diffusion. In the last experiment, you used cubes of alkaline agar jelly and an indicator, so that you could measure the time taken for an acid to diffuse through the cubes. Now you are going to plan how you can use this technique to investigate other factors. You may be able to do the experiment that you plan. 1. As well as surface area, diffusion is also affected by temperature, concentration gradient, distance. Choose one of these variables to investigate. 2. The variable that you change in your experiment is called the independent variable. What is the independent variable in your experiment? 3. Think about how you can change your independent variable. Decide how many different values you will have. 4. The variable that you measure, as you collect your results, is the dependent variable. What is the dependent variable in your experiment? 5. Describe how you will measure your dependent variable. 6. It is important that no variables, other than the one you are investigating, affect your experiment. Which variables will you standardize in your experiment? How will you do this? 7. Write a short, clear description of how you will do your experiment. 8. Draw a results chart that you could use to fill in your results. Include headings, but do not put in any results. 9. Predict what you would expect to find in your experiment. Questions. 5. Using what you know about how diffusion happens, explain each of these statements. A. Diffusion happens faster at higher temperatures. B. The greater the difference in concentration between two solutions, the faster diffusion happens. C. In a kidney dialysis machine, many small tubes containing dialysis fluid are used, rather than one big one. D. In a kidney dialysis machine, the membrane separating the blood and the dialysis fluid is very thin. 3.2 Osmosis. Water is one of the most important compounds in living organisms. It can make up around 80% of some organisms' bodies. It has many functions, including acting as a solvent for many different substances. For example, when we swallow food, some of it dissolves in water in the alimentary canal, where it can be acted on by enzymes which are also dissolved in water. The kidneys excrete urea dissolved in water to form urine. Substances are transported around the body dissolved in the water in blood plasma. Every cell in an organism's body has water inside it and outside it. Many different substances are dissolved in this water, and their concentrations may be different inside and outside the cell. This creates concentration gradients, down which water and solutes will diffuse, if they are able to pass through the membrane. It's easiest to think about this if we consider a simple situation involving just one solute. Figure 3.5 illustrates a concentrated sugar solution, separated from a dilute sugar solution by a membrane. The membrane has holes or pores in it which are very small. An example of a membrane like this is dialysis tubing. Figure 3.5. How osmosis happens. It is important to think about the diffusion of each kind of molecule separately. Water molecules are also very small. Each one is made of 2 hydrogen atoms and 1 oxygen atom. Sugar molecules are many times larger than this. In dialysis tubing, the holes are big enough to let the water molecules through, but not the sugar molecules. Dialysis tubing is called a partially permeable membrane because it will let some molecules through but not others. There is a higher concentration of sugar molecules on the right-hand side of the membrane in Figure 3.5, and a lower concentration on the left-hand side. If the membrane was not there, the sugar molecules would diffuse from the concentrated solution into the dilute one until they were evenly spread out. However, they cannot do this because the holes in the membrane are too small for them to get through. There is also a concentration gradient for the water molecules. On the left-hand side of the membrane, there is a high concentration of water molecules. On the right-hand side, the concentration of water molecules is lower because a lot of space is taken up by sugar molecules. Because there are more water molecules on the left-hand side, at any one moment more of them will hit a hole in the membrane and move through to the other side than will go the other way. Over time, there will be an overall, or net, movement of water from left to right. This is called osmosis. You can see that osmosis is really just a kind of diffusion. It is the diffusion of water molecules in a situation where the water molecules, but not the solute molecules, can pass through a membrane. Water potential. It can be confusing to talk about the concentration of water molecules, because the term concentration is normally used to mean the concentration of the solute dissolved in the water. It is much better to use a different term. We say that a dilute solution has a high water potential. A concentrated solution has a low water potential. In Figure 3.6, there is a high water potential on the left-hand side and a low water potential on the right-hand side. There is a water potential gradient between the two sides. The water molecules diffuse down this gradient, from a high water potential to a low water potential. Water potential and osmosis are of great importance to organisms because they affect how and when organisms take up and lose water. You will find out about the uptake and loss of water by plants in Chapter 8. The uptake of water by humans is described in Chapter 7. Figure 3.6. We can think of a water potential gradient as a slope. Water molecules have a tendency to move down the slope, from a high water potential to a low water potential. Questions. These questions are about the diagram in Figure 3.5. 6. What is the solvent in the sugar solution? What is the solute? 7. Explain why water molecules can move from one side of the membrane to the other, but sugar molecules cannot. 8 inches. Which direction is the net movement of water molecules? 9. Where does the energy come from to cause this movement? 10 inches. Figure 3.5. Which solution has the higher water potential? Explain your answer. Experimental skills 3.3. Using dialysis tubing to investigate osmosis. In this investigation, you will watch osmosis happen. You can also use the apparatus to investigate how differences in concentration of the solutions affect the rate at which osmosis happens. You will need the apparatus shown in Figure 3.7. A dropper pipette. A stopwatch. At least 2 different concentrations of sugar solution. Figure 3.7. Apparatus you will need for this investigation. It is best to do this experiment with a partner, as you often need at least 3 hands to do some of the steps successfully. Method. 1. Collect a piece of dialysis tubing. Put a few drops of water on it to soften it, and rub it gently between your fingers until it opens. Tie one end of it around itself in a tight knot. 2. Use a dropper pipette to carefully fill the piece of tubing with the most concentrated sugar solution. 3. Place a long, narrow glass tube into the dialysis tubing, as shown in Figure 3.7. Use some strong thread to tie the dialysis tubing very tightly around the glass tube. 4. Place the dialysis tubing inside a beaker of water, as shown in Figure 3.7. Support the glass tube using a retort stand, boss and clamp. 5. Mark the level of the liquid inside the glass tube. 6. Every 2 minutes, record the level of liquid inside the glass tube. 7. You can now try repeating your experiment, but this time use a different concentration of sugar solution. This is quite tricky to do, because you will need to take your apparatus apart to change the sugar solution. Record your results using the new sugar solution. 8. On a large sheet of graph paper, draw a line graph for your first set of results. Remember that the independent variable goes on the x-axis, and the dependent variable goes on the y-axis. 9. If you were able to repeat the experiment with a different sugar solution, plot another graph on the same pair of axes. Questions. 1. Explain why the liquid moved up the glass tube. 2. Use your graph for the first set of results to work out the mean rate at which the liquid moved up the tube, in millimeter per second. 3. If you were able to repeat the experiment with a different sugar solution, compare the two sets of results. Can you explain any differences between them? 4. Imagine you are able to use dialysis tubing with ridges on it, instead of a smooth surface. Predict how this might affect your results. Explain your prediction. 5. Suggest how you could modify this experiment to find out how temperature affects the rate of osmosis. Predict the results you would expect. Osmosis and animal cells. You have seen that osmosis happens when two solutions are separated from each other by a partially permeable membrane. The membrane lets water molecules through, but not the other molecules that are dissolved in the water. Cell membranes are partially permeable. They let water through easily, but other molecules and ions often cannot get through. This means that osmosis happens across cell membranes. Figure 3.8 shows an animal cell in pure water. The cytoplasm inside the cell is a fairly concentrated solution. The proteins and many other substances dissolved in it are too large to get through the cell membrane. However, water molecules can get through the membrane. Figure 3.8. If an animal cell is placed in pure water, water enters the cell by osmosis. The cell swells and bursts. If you compare this situation with Figure 3.5, you will see that they are similar. The dilute solution in Figure 3.5 and the pure water in Figure 3.8 are each separated from a concentrated solution by a partially permeable membrane. In Figure 3.8, the concentrated solution is the cytoplasm and the partially permeable membrane is the cell membrane. Therefore, osmosis will occur. Water molecules will diffuse from the dilute solution into the concentrated solution. As more and more water enters the cell, it swells. The cell membrane has to stretch as the cell gets bigger, until eventually the strain is too much, and the cell bursts. Figure 3.9 shows an animal cell in a concentrated solution. If this solution is more concentrated than the cytoplasm, then water molecules will diffuse out of the cell. Look at Figure 3.5 to see why. As the water molecules go out through the cell membrane, the cytoplasm shrinks, so the cell gets smaller. Figure 3.9. If an animal cell is placed in a concentrated solution, water leaves the cell by osmosis. The cell shrinks. Osmosis and plant cells. Plant cells do not burst in pure water. Figure 3.10 shows a plant cell in pure water. Plant cells are surrounded by a cell wall. This is fully permeable, which means that it will let any molecules go through it. Figure 3.10. If a plant cell is placed in pure water, water enters the cell by osmosis. The cell swells but does not burst, because of its strong cell wall. Although it is not easy to see, a plant cell also has a cell membrane, just like an animal cell. The cell membrane is partially permeable. A plant cell in pure water will take in water by osmosis through its partially permeable cell membrane in the same way as an animal cell. As the water goes in, the cytoplasm and vacuole will swell. However, the plant cell has a very strong cell wall around it. The cell wall is much stronger than the cell membrane, and it stops the plant cell from bursting. The cytoplasm presses out against the cell wall, but the wall resists and presses back on the contents. In a plant leaf, cells that have plenty of water are all in this state. In each one, the cytoplasm presses out on the cell walls. The pressure of the water pushing outwards on the cell's walls keeps the whole group of cells strong and firm. This helps to support plant leaves. A plant cell in this state is rather like a blown-up tire, tight and firm. It is said to be turgid. The pressure of the water pushing outwards on the cell wall is called turgor pressure. Turgor pressure helps a plant that has no wood in it to stay upright, and keeps the leaves firm. Plant cells are usually turgid. Now imagine that a plant cell is placed in a solution that is more concentrated than its cytoplasm. Water leaves the cell by osmosis. The cytoplasm and vacuole shrink. But the cell wall is strong and stays in position. The cytoplasm therefore pulls away from the cell wall. This pulls the membrane away from the cell wall, too. The cell now looks like the diagram in Figure 3.11 and the photographed cells in Figure 3.12. Figure 3.11. If a plant cell is placed in a concentrated solution, water moves out of it by osmosis. The cytoplasm and vacuole shrink, and the cell membrane pulls away from the cell wall. Figure 3.12. These onion cells have been placed in a concentrated solution. The cytoplasm has lost so much water, and shrunk so much, that it has pulled away from the cell wall. When a plant cell loses a lot of water, it also loses its turgor pressure. The contents of the cell do not push outwards on the cell wall, so the cell becomes soft and floppy. It is said to be flaccid. If the cells in a plant become flaccid, the plant loses its firmness and begins to wilt. When the cell membrane tears away from the cell wall, the cell is said to be plasmolyzed. Plasmolysis can kill a plant cell because the cell membrane is damaged as it tears away from the cell wall. Questions. 11. Copy and complete this sentence. When an animal cell is placed in pure water, water the cell by. Through the partially permeable cell. 12 animal cells burst if they are placed in pure water, but plant cells do not. Explain why. 13. Here are some descriptions of what happens to a plant cell when it is placed in a concentrated solution. They are in the wrong order. Write the descriptions in the correct order. The cytoplasm and vacuole shrink. The cell membrane is pulled away from the cell wall. Water moves out of the cell through the partially permeable cell membrane. 14. Look at Figure 3.11. What fills space X? Explain your answer. 15. A group of plant cells has been placed in a concentrated solution, and the cells are plasmolyzed. Predict what will happen if the cells are now placed in pure water. Explain your answer, using the term water potential. Experimental Skills 3.4. Investigating the effect of osmosis on potato strips. In this investigation, you will place pieces of plant tissue into different solutions and measure the effect on their length. You will need a large potato, a sharp knife or other way to peel the potato, a cork borer to cut cylinders from the potato, a ruler to measure in millimeter, 3 containers such as small beakers, some distilled water, a dilute sugar solution and a concentrated sugar solution. Method 1. Peel the potato. Use a cork borer to cut 6 cylinders from the potato. Cut the ends from each cylinder so that each one is the same length. 40 millimeters is a good length to use. Record this length. If you do not have a cork borer, you can cut rectangular pieces from the potato instead. Figure 3.13. You can cut either cylinders or rectangular blocks from the potato. 2. Pour the distilled water into 1 beaker, and the 2 sugar solutions into the other 2 beakers. Label each beaker with the concentration of the solution. 3. Place 2 potato strips into each liquid, making sure they are completely covered. 4. Leave the potato strips in their liquids for about 20 minutes.